afternoon, and welcome to the Alamo Breast Cancer Advocate Chat. My name's Mary Jimenez. I'm a healthcare executive leader in the biotech and diagnostic industry. I am co-founder of iConsult, where I'm currently consulting for several startup companies, HealthGates, Sizzle Biotechnology, and Scientra. I also serve on ASCO's Women Who Conquer Cancer Committee, the San Antonio Medical Foundation's Community Health and Education Committee, the UT Mays Cancer Centers Advisory Board, the ThriveWell Board, and Alamo Breast Cancer Foundation's Advocate Committee. Joining me today here is Dr. Kristina Rinker. She is professor and associate head research in the Department of Biomedical Engineering at the University of Calgary and founder and chief scientific officer of Scientra. She received her PhD in chemical engineering with biotechnology minor from the North Carolina State University and was previously an assistant research professor in biomedical engineering at Duke University. Dr. Rinker is also jointly appointed in the Cummings School of Medicine in the Department of Physiology and Pharmacology and is a member of the Arnie Charbonneau Cancer Institute, where she led the Early Cancer Detection Initiative from 2017 to 2020. She is also the founder and on the board of directors for a nonprofit, BioHub X, a catalyst for life science ecosystem in Alberta that supports companies scaling and expanding into global markets. Dr. Rinker's research is in the areas of diagnostic and therapeutic platform development. Her research has been supported through many organizations such as Alberta Cancer Foundation, the NSERC, NIH, and Whitaker Foundation. In 2024, she received a U.S. Department of Defense Breast Cancer Research Program Award with Dr. Kristina. Her team won the Project Achievement Award for the Scientra DX Early Cancer Detection Blood Test Platform from the Association of Professional Engineers and Geoscientists in Alberta, and the Innovation of the Year Award in 2023 from the BioAlberta Industry Trade Organization. Please join me in welcoming Dr. Rinker. She will be discussing some of the gaps and opportunities in early breast cancer detection.
Thank you, Mary. Thank you for the invitation to talk with you today.
Wonderful, Dr. Rinker. Well, before we get started and dive into some of the wonderful research that you've been working on, can you please share why early cancer detection is so critical?
So as we know, breast cancer affects many women. It's the most diagnosed cancer in the world, with 1 out of 8 women developing breast cancer in their lifetime. But what's less well known is that breast cancer is the leading cause of disease-related death for younger women 35 to 49. Actually, 20% of breast cancers almost 20% of breast cancers are in women under the age of 50. And this percentage is increasing. So breast cancer is a large problem. We've been looking at breast cancer diagnosis. Now, what's important within breast cancer diagnosis? If we're able to detect breast cancer at the earliest invasive stages, there's a 99% survival rate, 5-year survival rate. So it's really important to not only detect breast cancer, but also detect it at early stages. So in terms of how we're doing in breast cancer diagnosis, this is one particular study that was looking at the earliest invasive stages and the percent of women that have their breast cancer detected at those stages. And at best, we're doing about 50% of breast cancers for older women are detected at the earliest invasive stages. So there's lots of room for improvement here. And as you can see, for younger women, we're really diagnosing earlier at the earliest invasive stage, about a quarter. So there's lots of room for improvement here. And this is also a global priority. So the World Health Organization launched their Global Breast Cancer Initiative, and the first pillar of that is to increase early stage detection. increase the percentage of women around the world that get that early stage detection.
I totally agree, Dr. Rinker. I mean, can you share with us right now, what are the current diagnostic tools, being used for breast cancer detection, and what are some of the gaps? In a previous advocate chat with Dr. Peter Kuhn, he had mentioned that mammography doesn't work for everyone. So perhaps maybe you can tell us why.
Okay, so breast cancer has been detected primarily in more recent decades through the use of mammography, or most broadly with mammography-based screening. That's been increasing over the past decades and increased the early detection of breast cancer. Some women are also detected using MRI. But as we look to the future, as we look to how can we improve early stage diagnosis for women, we need to look at where the technologies are working best and where there's gaps and opportunities. I'll share a slide here to highlight for mammography in particular, what we're seeing in terms of some gaps. So for mammography, mammography is an X-ray image, and it's going to capture cancer as a white object. So white shows up very well against a black background or a dark background. And for women with low tissue density in their breasts, this leads to being able to detect tumors when they're very small. However, as tissue density increases, there's increased whiteness to the image, and that can mask the presence of a cancer, particularly at the smaller sizes. So as we look at the sensitivity of mammography, which is the ability of mammography, a screening mammogram, to detect the presence of cancer, it's affected by tissue density as well as the size of the tumor. So mammography sensitivity is shown in this chart here, which is in a review paper that we've published this year in 2024. We see that mammography sensitivity tends to decrease with increasing breast density. And this affects about half of women have C or D breast density. So it's a lot of women that are affected by this to varying degrees. This is also skewed towards younger women. So here underneath, it shows percent of women under age 45 with increased tissue density. And we see that it's skewed to the right there in terms of more C and D density for younger women. So breast density is more of an issue as you go to younger women. A higher percentage of the population has dense tissue. So as as we look to increase early stage detection, this is one of the gaps and opportunities for new technologies.
Yeah, definitely. And, we need more tools, and perhaps maybe you can share with us just a little bit, as I'm thinking about this. There are many diagnostic screening tests entering the market. Perhaps maybe you can share how blood tests could potentially help address these gaps in the future, and, why it could be a potential good solution.
Sure. So as we look at early breast cancer detection, it's not affected only by the performance of the screening technologies that are used. It's also affected by women actually participating and using the screening, so participating in screening programs, as well as having easy access for scheduling and transportation to the place where the screening is available. So there's a variety of aspects that we can look to improve to increase early stage detection in all women. So as we look to that, a blood test as a platform has the potential to really supplement our current imaging technologies, and that a blood sample can be easy to collect, can be collected potentially remotely, possibly as part of a routine doctor's visit. So collecting a blood sample, screening, a platform like that could make screening more accessible and could increase participation in screening programs, and also complement imaging-based screening and imaging-based detection. So what happens now is if you have a screening image that's concerning, or you see something there, it has a high score, you'll end up getting most likely followed up with additional diagnostic screening, diagnostic mammograms, ultrasound, MRI. And if there's still something there concerning, you'll get a biopsy and then pathology on that sample. And approximately 80% of those biopsies are negative for cancer. which is good, but this is a pretty inefficient process. So as we look to the future, how can we make this process more efficient to actually get the right technologies for the right population to enable some efficiencies there, as well as enable that earliest invasive stage diagnosis?
Yeah, I would agree. You want to catch that cancer early, but you also don't want to be sending patients, forward with more invasive tests if you don't need to. And if there's a way to look at that aspect of diagnostics, it would be huge for women. And it sounds like it can address an unmet need. So thank you for sharing that. Can you talk to us a little bit about the new platform that you've developed for early detection, and tell us about how this technology came about, some history around how it came about? Thank you.
Yeah, sure. So my research program has been traditionally based on cell-based testing, cell-based models for biomarker discovery, drug target discovery, drug testing in cardiovascular disease, inflammation, other areas. So about a decade ago, a group came from South Korea, and we began a collaboration around lab test development for breast cancer. Because at that time, I hadn't had a mammogram, and I'm like, okay, well, why do we need it? Oh, wait, we do have techniques like mammogram, but there are gaps, and we need to improve that. And a lab test is a good way to potentially address this problem. So we worked on this lab test development for the past decade, developing biomarkers, validating biomarkers, and then developing a platform for this that enables robust testing. So from the beginning, we had a different mindset. I guess we were looking at the technologies that were available. We're looking at separating into plasma, identifying biomarkers different ways. But when we thought about how to scale it up, how to develop a test that could be used around the world and run millions of tests, we thought about it and said, well, if we could use a whole blood sample, that would really make processing more easy. And so we started with a whole blood sample rather than going with fractionation of a blood sample like many of the other technologies under development or in research now are doing. And with the whole blood sample, there's a lot of signal there. So the trick is to actually find the specific signals that you're looking for out of that sea of signals. And we were able to develop a discovery platform that identified a panel of RNA markers. And then we used a detection technology that was readily available. And we selected PCR technology because it was readily available around the world. Plus, it actually amplifies the signal. So it enables us to measure even smaller amounts of material. So with this development, we also incorporated a software package, and the software incorporates machine learning based algorithms to make the test robust and specific, and to give it a lot of breadth so that it can be continuously improved over time.
Wow. So it sounds like your test is starting to look at high-risk patients in breast cancer and looking at a broader population, young women, with your study. I know that when you start looking at broader populations, can you share the different aspects of validating a platform like this, and what are the important things to look for? How does this compare to other tests in the market as well?
Sure. So in terms of validating the test, I mentioned a little, I'm going to focus really on the screening test, or the early stage detection tests, where for a technology, you typically do the analytical validation, meaning does it measure what you want it to measure, as well as not measure what you don't want to measure. And then the clinical validity, such as, is it working in the population to pick out the disease or no disease? I mentioned a bit earlier about mammography and sensitivity. So there's two measurements really in diagnostics that I'll focus on here, which is sensitivity and specificity. Sensitivity is the ability to detect the cancer, the breast cancer, pick out the breast cancer as well, whereas the specificity is the ability to pick out the negatives well. And those are going to be dependent on your cohort. So I'm going to highlight that with a slide that we've previously presented on our platform. So for our new platform blood test, we began a study in 2018 on the clinical validation of the locked down product version of the test in an accredited laboratory. And for this study, it's blinded, meaning that the test was already locked down and the processing or running of the test was blinded to the patient outcomes. We didn't know who was positive or who was negative for the presence of breast cancer. So in performing this, our study involved women ages 25 to 80, and the accuracy of the test came out to 92% with a specificity of 94% and a sensitivity of 79%. This test performed even better for young women. So you see to the right there, younger women ages 25 to 49, where we're seeing higher sensitivities and specificities even for this population. So when we're talking about sensitivity and specificity, this will depend on the cohort, as I mentioned, and 84% of these participants had breast cancer at either stage 1 or stage 2. And as part of this analysis, we were able to show, importantly, that the test was able to detect early stage cancers, and including tumors under 10 millimeters. So when we're looking at the competitive landscape of what's out there, there's lots of tests that are out there. And one of the things to look at as you're evaluating tests and literature is, what stage of the tests on and what populations are their clinical validations on. We see a lot of technologies involving circulating tumor DNA or markers such as that, cell-free DNA. And breast cancer is not a big shedder of material into the bloodstream. And so that has made a lot of the technologies that are based on the shedding into the bloodstream to be, it's harder to detect at the earliest invasive stages when the tumors are really small, because really you're looking for a needle in the haystack. Whereas with our approach and our new platform, again, we have a whole blood sample, about a teaspoon, so it's not a lot of blood that's needed for the test. But we're able to detect out that signal from a larger group of signals and do it accurately and robustly. The other thing I'll mention is the test does have CE marking and runs under accreditation. So we do have, with this technology, it's been developed over the past 10 years, so a fair bit of validation associated with that.
Wow. So this is exciting news for looking at smaller tumors, which means earlier stage, which can impact overall survival. So, I think the challenge now is to better understand the technology, so we can harness it in a way that we know can save lives. And congratulations to you and your team, because it sounds like you're heading in that right direction. So before we close, I'd love to just ask you, what do you think we can do to improve early detection in women? What else could we do?
Yes, so I think it takes a community. It takes all of us to participate, the patient advocates, the clinicians, the healthcare systems, the innovators, to work towards further clinical studies and validation of technologies. How do we implement these technologies in clinical care pathways in the ways that are going to be most beneficial to the women and increase early detection? How do we move the bar to enable us to have access to new technologies while maintaining the safety of the individuals? So this is really a community participation in this to bring these advances into early cancer detection, because every woman is different. There's a different genetic profile, different life situation, different geographies. And we need to have more options that are accurate and effective and cost effective to enable those solutions to be available for women and to increase early stage detection for all women.
Well, thank you so much, Dr. Rinker. Is there anything additional you would like to add to this conversation today?
No, I don't think so. I really appreciate the time today, and thank you very much.
You're welcome. So thank you, Dr. Rinker, for sharing some valuable information with us today at the Alamo Advocate Chat. We'd like to thank all the patients who have continued to contribute to research and advance diagnostic tools for early detection. Thank you.
Thank you.